We are on the subject of microbiology is irreducibly complex and demands an intelligent creator. And we scrolled on down to something that I hadn't thought about, and I added this to this series. Let me just go to the safe place here. We are driven by science and logic to admit that life in any form can have its source only in a God who is independent of the material universe. That there cannot be more than one source is proved by the uniformity and universality of the language. DNA, we're talking about the language of DNA, how things reproduce. DNA, of course, does not understand the information encoded into it. It is a mechanism. It didn't evolve. It is a mechanism built and programmed by the originator of the encoded language to follow his instructions automatically. We already looked at the beginning of this study, and it could not have evolved this far, far too complex, just the DNA alone of a creature, a live creature. And the most complex mechanism built by DNA, of course, is the human brain. More advanced than any computer yet built by man, it contains some 100 billion nerve cells connected by 240 miles of nerve fibers involving 100 trillion connections. That's in your brain. Tell me that evolved. For all of its complexity, the brain no more originates or understands what it is doing than does DNA. The brain does not originate thoughts. If it did, we would have to do whatever our brains decided. On the contrary, we, the real persons inside, do the thinking and deciding, and our brains take these non-physical thoughts and translate them into physical actions through a connection between the spirit and body that science can't fathom to this day. Tell me that evolved. You need something more advanced than the human brain, even, and the actions of the humans with that brain in order to create humans more intelligent than humans. That creator has to be in order for humans to exist. Wilder Penfield, one of the world's leading neurosurgeons, describes the brain as a computer programmed by something independent of itself, the mind. It's not you don't say, I went to my doctor's office and he operated on my mind. Even psychiatrists don't do a good job of that. Science cannot escape the fact that man himself, like his creator, must be a non-material being in order to originate the thoughts processed by the brain. But man did not originate thought itself. He did not create himself, nor give himself the capacity to think. So here's a statement in caps. Evolution does not have a chance to explain how the blood coagulating system works. So let's go to this second subject. In Michael Behe's book, Darwin's Black Box, he says, is it possible that this ultra-complex system could have evolved according to Darwinian theory? Talking about the blood coagulating system. I selected this one particular system because it's more real to me. Should I cut myself? How do I get saved from bleeding to death? And then how do I get saved from the heart getting imploded by the blood coagulating system because it never stopped? The observation of split genes, wherein similarities were observed in the genetic structures of different parts of biological systems, led to the hypothesis that perhaps new proteins could be made by shuffling the DNA fragments of genes that code for parts of old proteins, much as cards can be picked from several piles to give a new arrangement. To support the hypothesis, advocates point to similarities in the amino acid sequences and shapes of discrete portions called domains of different proteins. The proteins of the blood coagulation cascade are often used as evidence for shuffling. Some regions of cascade proteins coded by separate gene pieces have similarities in their amino acid sequences with other regions of the same protein. That is, they are self-similar. Also, there are similarities between regions of different proteins of the cascade. For example, proconvertin, Christmas factors, Stewart factor, and prothrombin all have a roughly similar region of their amino acid sequences. Additionally, in all those proteins, the sequence is modified by vitamin K. Furthermore, the regions are similar in sequence to other proteins not involved with blood coagulation at all, and that are also modified by vitamin K. So you can build a chance here, a chance that maybe this could happen. Let's keep reading. The sequence similarities, go up a little bit. The sequence similarities are there for all to see and cannot be disputed. By itself, however, the hypothesis of gene duplication and shuffling says nothing about how any particular protein or protein system was first produced, whether slowly or suddenly, or whether by natural selection or some other mechanism. Remember, a mousetrap spring might in some way resemble a clock spring, and a crowbar might resemble a mousetrap hammer, but the similarities say nothing about how a mousetrap is produced. That's the point we were making with bacteria that are similar and have similar components, but have different functions for some of those components. In order to claim that system developed gradually by a Darwinian mechanism of evolution, a person must show that the function of the system could have been formed by numerous successive slight modifications within an acceptable time frame. The explanation is seriously inadequate because no reasons are given for the appearance of the proteins. No attempt is made to calculate the probability of the protein's appearance precisely the right time and place, and no attempt is made to estimate the new protein's properties. It's like you go in a closet, at least bring a flashlight. The first thing to notice is that no causative factors in the evolutionary models proposed are cited. Thus, tissue factors appears, fibrinogen is born, antiplasmin arises, TPA springs forth, and cross-linking protein is unleashed, and so forth. What exactly, we might ask, is causing all this springing and unleashing? A step-by-step Darwinian scenario involving the undirected random duplication and recombination of gene pieces is in view, but consider the enormous amount of luck needed to get the right gene pieces in the right places at the right time. Eukaryotic organisms have quite a few gene pieces, and apparently the process that switches them is random. So making a new blood coagulation protein by shuffling is like picking a dozen sentences randomly from an encyclopedia in the hope of making a coherent paragraph. The time it takes that is beyond the age of the universe. Just for the blood coagulating system. What do you do about everything else of the human body? They all have to work in coordination one with another. So furthermore, the Darwinian scenario typically does not go to the trouble of calculating how many incorrect, inactive, useless, variously shuffled domains would have to be discarded before obtaining a protein with, say, TPA-like activity. To illustrate the problem, let's do our own quick calculation. Consider that animals with blood clotting cascades have roughly 10,000 genes, each of which is divided into an average of 3 pieces. This gives a total of about 30,000 gene pieces. TPA has 4 different types of domains. By variously shuffling, the odds of getting those 4 domains together is 30,000 to the 4th power, which is approximately 1/10 to the 18th power. Now, if the Irish sweepstakes had odds of winning of 1/10 to the 18th power, And if 1 million people played the lottery each year, it would take an average of about 1,000 billion years before anyone, not just a particular person, won the lottery. 1,000 billion years is roughly 100 times the current estimate of the age of the universe. There you go. And we haven't even gotten a coherent working blood clotting system within that human. The same problem of ultra-slim odds would trouble the appearance of prothrombin, fibrinogen, and plasminogen, proachlorine, and each of the several proposed rearrangements of prothrombin. Unfortunately, the universe doesn't have time to wait. Already, we've got far greater time that the universe took to form, according to evolutionists. The second question is to consider is the implicit assumption that a protein made from a duplicated gene would immediately have the new necessary properties. Thus, we are told by the Darwinian model type, tissue factor appears as the result of the duplication of a gene for another protein. Looking around, oh, in that dark closet without a flashlight. If a factory for making bicycles were duplicated, it would make bicycles, not motorcycles. That's what is meant by the word duplication. A gene for a protein might be duplicated by a random mutation, but it does not just happen to also have sophisticated new properties. Since a duplicated gene is simply a copy of the old gene, an explanation for the appearance of tissue factors must include the putative route it took to acquire a new function. This problem is discreetly avoided. See, the Darwinian model runs into the same problem in the production of prothrombin, a thrombin receptor, antithrombin, plasminogen, antiplasmin, and proachlorine, Stewart factor and proconvertin, Christmas factor, antihemophilic factor, and protein C, virtually every protein of the system. So the third problem in the blood coagulation scenario is that it avoids the crucial issues of how much, how fast, when, and where. Nothing is said about the amount of clotting material initially available. The strength of the clot would be formed by a primitive system, the length of time the clot would take to form once a cut has occurred, what fluid pressure the clot would resist, how detrimental the formation of inappropriate clots would be, or 100 other such questions. And when it stops clotting, because if it keeps on clotting, the human will die. The absolute and relative values of these factors and others could make any particular hypothetical system either possible or, much more likely, wildly wrong. For example, if only a small amount of fibrinogen were available, it would not cover a wound. If a primitive fibrin formed a random blob instead of a meshwork, it would be utterly unlikely to stop the blood flow. If the initial action of antithrombin were too fast, the initial action of thrombin too slow, or the original Stewart factor or Christmas factor or antihemophilic factor bound too loosely or too tightly, or if they bound to the inactive forms of their targets as well as the active forms, then the whole system would crash. No more blood clotting system. How many times do you have to do that? After 1,000 times of universes existed and went out of existence, we still don't have a blood clotting system. At no step, not even one, does any of the Darwinian models give a model that includes number or quantities. Without numbers, there is no science. When a merely verbal picture is painted of the development of such a complex system, there is absolutely no way to know if it would actually work. When such crucial questions are ignored, we leave science. And they do that. Yet the objections raised so far are not the most serious. We go on that next time.